Good morning, good afternoon, good evening, everyone, and welcome to this webinar on the effects of plastic pollution on Arctic animals. My name is Rene Larsen from the AMAP Secretariat, and I will introduce the webinar today. Before we go into the actual webinar, just a few words about AMAP. It was established in 1991, and it's a working group of the Arctic Council, became part of the family in 1996. We have delegates from 8 Arctic states, from 6 permanent participants, which is the name that we use for indigenous organizations. We have observers from many nations. We have expert groups covering approximately 800 experts, and our secretariat is located in Tromsø in Norway. The mandate of AMAP is to monitor the levels of and assess the effects of anthropogenic pollutants in all components of the Arctic environment. We also have the mandate of assessing the effects of climate change on Arctic ecosystem, and then finally an understanding of the impacts on human health and the effects of multiple stressors on human health. As one of its mandates, as part of the mandate, AMAP works on litter and microplastics pollution, and we have established a litter and microplastics expert group, and that has published monitoring guidelines, monitoring plan, and monitoring guidelines in 2021. Then we are currently working on a scientific report on effects of plastic pollution on Arctic biota. It's peer-reviewed and will be published in summer 2025. And then recently, in May 25, we released a so-called summary for policymakers on the effects of plastic pollution on Arctic animals. And it's mainly, and it's the work, these two pieces of work that we will present to you today. The program is, as you can see here, a small introduction by me, and we have 4 speakers. Eivind Farmen will offer summary and recommendations, and then finally there will be room for questions and answers. I should kindly ask you to wait with your questions and comments. So we've completed all presentations. We'll find a mechanism where the audience you can raise your hand and ask your questions. We will not monitor the chat through this, so if you have questions or comments, please raise your hands towards the end. A few words on the background here. Multiple studies have documented the presence of plastics both in the abiotic environment, in the Arctic, and of the Arctic fauna. Plastic pollution is a complex problem because plastic varies in size, morphology, and chemical constituents. AMAP has defined a series of priority indicators for circumarctic monitoring, and these indicators include beach litter, microplastics in water and sediment, and ingested plastics in seabirds as our first priority indicators. Negative effects from plastic pollution in the Arctic have been considered in some scientific studies. However, there are no common indicators for these effects that have been proposed or implemented to date in the Arctic region or elsewhere to specifically measure biological effects of plastic pollution. The work presented today, as I said, is based on a report that we will soon publish. It summarizes the state of knowledge on the negative biological effects of plastic litter and microplastics and their additives on Arctic animals, and it will analyze the current gaps in our knowledge in this field. Plastic pollution has a global interest, and the United Nations endorsed a resolution to develop a legally binding agreement to end plastic pollution, and there is an intergovernmental negotiating committee, INC, that has been established to work towards the global treaty. It was expected to complete its work by 2024, but is still working, and the next session in that series is in August this year. But without further introduction, I would like to present our first speaker, Ingrid Pollet, who will speak on biological effects of litter entanglement. Ingrid is a Canadian ecological consultant with expertise in seabird ecology and conservation. She collaborates with the Biota College, with Acadia University, and the Environment and Climate Change in Canada. Her research projects focuses on seabird survival, movement ecology, contaminants, and bycatch. Her recent work addresses the impacts of plastic pollution on marine wildlife, including entanglement. So please, I'll stop sharing, Ingrid, and over to you.
Hello, everyone. So it won't come as a surprise to any of you that plastic is present everywhere, from pole to pole, from the bottom of the ocean to the highest mountain top. And just like plastic is present everywhere, entanglement of wildlife in litter is also a global issue. In the Arctic alone, entanglement has been recorded in 12 species of marine mammal, 24 seabird species, and 3 species of terrestrial mammal. It has also been recorded in fish, in coral, and in crustaceans. The litter involved in entanglement can be anything from discarded fish gear, which could be commercial or recreational, and consumer plastic. At the individual level, the effect of entanglement are often clear and observable, and the impact depends on which body part is entangled. Entanglement can decrease mobility, which increase vulnerability to predators. It can cause wound and laceration, leading to infection. And ultimately, entanglement can result in starvation and death. But while the individual level are obvious, what do we know about population level impact? So I'm going to take seabird as an example because that's what I studied most. And for most arctic breeding species, we have a decent population data, and we also have population trends for most species. But how much of any population decline is due to entanglement, we just simply don't know. So to assess population impact of entanglement, we need to access entanglement data. And so part of this sub-working group was to search for such database. Relevant entanglement data can be found in a variety of databases, such as Pitchbird and Entanglement Program, which is based on standardized protocol with monthly survey. There's also citizen science platforms such as eBird and iNaturalist, where people record their opportunistic observations. And then there's plastic monitoring tool, such as the Marine Debris Tracker, where the main goal is to report plastic litter, but some databases keep track of entanglement event that they observe. So as you can see, each database differ in geographical scale from local, regional, or global, in the volume of data provided by those databases, from tens of entry to millions of entry, depending on the number of year and the number of contributor, but also has a different quality control. Not all databases are harmonized, and some probably don't have much in terms of quality control. And then the frequency of observation from regular survey to opportunistic sighting. Finally, some of those databases are freely accessible online, while others require permission to access the data. And most databases are only in English, and some include images to validate the observation. So in summary, it's very difficult to harmonize data from all those different sources. So basically what we want to know is the total mortality of from entanglement, which is the bigger circle. And let's be honest, it's just not possible to get this value. But we can have access to other estimates. But from each step, from the smaller circle to the big circle, we are losing some certainty in our estimate. So the most precise value are represented by the small circle. And those are entanglement animals that are reported in accessible database following a standardized protocol. Then we have entangled animal reported using all the different databases that we found. And the quality control is not as strict as the previous circle, so we are already losing some certainty. Then we have entanglement animal that could be reported if standardized protocol were carried out across the species range, which is very difficult to get. And in the Arctic, the challenges of finding population impact of entanglement is even greater than some other places because the human population is very low, which means that there are fewer observers to report entanglement and that entanglement proofs could disappear before somebody could witness it. There's also no standardized database across a circumpolar Arctic, and most existing databases are local or regional, making large-scale analysis difficult. So at this point, estimating the population level impact of entanglement in the Arctic is not really feasible. However, we do have some recommendations to improve the situation. And we decided that instead of creating yet another database, we suggest a cross-promotional approach, encouraging existing database to collect and share as much standardized information as possible. And the essential information should include the date and the location with the coordinate of the encounter, the species name, or at the very least, the taxonomic group, the status of the animal, is it dead or is it alive, and its age, is it an adult or is it an immature, and the number of individuals involved in one given event. It should also indicate if the entanglement has been reported in another databases to avoid duplicate content. And finally, the data should include the litter type that the animal is entangled in. Is it a fishing net? Is it a rope? Is it a plastic bag? Or what is it? And finally, all databases reporting wildlife entanglement should be made available and accessible to all. Thank you.
Thank you very much, Ingrid. And again, any question comes from the audience, there'll be a session towards the end. I would like to introduce Amy Lusher, who will talk to us about physical effects of plastic ingestion. And Amy is a senior research scientist at the Norwegian Institute for Water Research. She's a marine biologist who has focused her career on the topic of microplastics and litter pollution. And her research currently focuses on the necessity of best practice methods to understand distribution, interactions, and potential effects of plastics in the environment. So please, Inge.
I'm going to assume that you can see my screen okay. Thanks for the introduction, Jan. So I'm presenting this work on behalf of the sub-working group that was led by Mark Mallory. And we focused on looking at the physical effects. As just briefly introduced by Ingrid, there are many interactions between animals and plastics, and many extensive reviews have been carried out on how animals are affected, including ingestion, entanglement that's just observed it and habitat damage. But we're really going to just focus on entanglement here, and specifically the physical damage which is caused by ingested plastic to the digestive systems of Arctic wildlife. So quite a specific topic, but it's not so easy to explain the outcomes, as you'll see on the next couple of slides. So when plastic ingestion occurs, there's many things that can happen. In the best case scenario, it passes straight through the body, it's eliminated as solid waste, or it can also be regurgitated. But it can also physically reduce or block the passage of foods. It can irritate and penetrate or even cut surrounding tissues in the digestive tract. And these lesions can increase any risk of infection to the biota. This can lead to starvation or reduced feeding with reduced nutritional food intake, and that can also lead to reduced body condition, and in some cases this may lead to mortality. So what do we know about this in the Arctic wildlife? Most species in the Arctic have actually not been examined for plastic ingestion. We have a lot of information for marine species, including seabirds, invertebrates, fish, and mammals. And to look deeper into this, I suggest to look into the AMAP previous work that we've done, and also our publications that was produced afterwards. We also highlighted that there is no terrestrial reports on invertebrates, and there are a few reports for terrestrial birds and mammals and freshwater fish. When it comes to physical damage, in most instances, what we're seeing is the presence of the plastic. So we know that the plastic's there, but there's no systematic approach to understanding or recording evidence of physical damage. So to give two examples, in marine mammals, we see low numbers of debris being detected inside stomachs, but there's no evidence of physical damage. In seabirds, none of the studies report mortality resulting from plastic ingestion, although one study did highlight the potential for physical damage that could have been caused by the plastic particles themselves. And this was particularly a hole inside the proventricular of the fledgling, and also perforations in the intestinal wall. Additional physical damage that we could consider, that at the moment we have no evidence from the Arctic, could be novel damage such as shifts in gut microbiomes, pathogenic and antibiotic-resistant bacteria, as well as extensive scar tissue in digestive tracts. There's been evidence in animals outside of the Arctic, but we currently do not have any evidence from the Arctic region. In our understanding of plastic physical effects in Arctic biota, we're very much limited by the spatial and taxonomic gaps in the knowledge. We're also limited to access to dead animals. There could be difference between beached, by-caught, and harvested animals. And also, it's important to consider the sample size that might be needed in order for us to detect change. So, to put it another way, look at it from an ethical situation, should really get out killing animals to see if they're affected by plastic pollution. Actually, they could be okay with the plastic that they've still got in their guts. So it's something that we've posed as a topic we want to look further into within our sub-working group. So our main knowledge gaps at the moment, as already highlighted, are the terrestrial biota, freshwater fish, marine invertebrates, and marine fish. So the recommendations we highlighted were to develop a pan-Arctic marine mammal stranding network that could allow us to use harmonized sampling protocols and data reporting so we can actually assess damage rather than just looking at the presence. And we could do this by aligning with existing global programs. One example is the development of indicators under the Marine Strategy Framework Directive that are looking at harm caused by plastic ingestion. So we suggested that we should be establishing and enhancing sampling programs with the support of Indigenous or sports hunters, and also work with commercial industries, which would allow us to get sufficiently high sample sizes to look for, to detect a change. But I think the most important outcome that we had within the working group is that the physical damage should probably be lower priority when compared to other impacts such as entanglement or the effects of chemicals. And we'll hear more about the effects of chemicals from Bonnie. So that's a very, very short summary. Didn't have too much to say, and I look forward to hearing any questions later on. Thank you.
Thank you very much, Amy. Your voice was a bit distorted during the presentation, but I hope all could hear it. But I think it was okay. I believe I understood everything there. So thanks a lot. Next, Tanja Kögel from Norway will be presenting on physiological effects from ingested micro and nanoplastics in Arctic animals. And Tanja is a PhD and scientist at the Institute of Marine Research in Norway. She has studied at the Johannes Gutenberg University of Mainz and the Ruprecht Karls University, Heidelberg, Germany, and she has a PhD from University of Bergen in Norway. Her background is in cell biology with focus on intercellular transport and hormone secretion. And since 2015, she's been working on contaminant mapping and monitoring, method development and effect studies, including microplastics, mainly in seafood organisms. She's currently one of the chairs of the ICES working group on marine litter. And with that, Tanja, please.
Thank you. So I will be presenting our gap analysis of our newest work on physiological effects from micro and nanoplastics in Arctic animals. And here we focus on microplastic and nanoplastic particles below 5 millimeters, because the larger plastic particles have been covered by the previous 2 chapters, which you just heard about. So this work builds on our previous product, the monitoring guidelines, and several published journal articles that grew out of that on occurrence of these microplastic particles in mammals, birds, invertebrates, and fish, respectively, and the methods that have been used. So what we wanted to do in this new gap analysis is now to combine the knowledge we have on the occurrence of the plastics with the effects of the plastics to see if we can say something about the risk of this pollution. Now, we try to include the entire body of literature in Arctic on animals which have an Arctic habitat. And that body of literature was not very large. It was 112 articles in total, of which only 26 were on effects. And there were no effect studies on Arctic mammals, set aside laboratory rats and mice. There were quite a high number of occurrence studies in birds, but no effect studies. And there were some effect studies in fish, and there were some more effect studies in invertebrates, even more than occurrence studies. So some details from this study show that occurrence in Arctic mammals was mostly on seals, walrus, and fin whales, and there is a lack on data on any land mammals. Besides the mice and rat. And we think it would be very interesting with more data, or with some data on reindeer or caribou, how they say in Canada, because they are also eaten. There was some evidence for translocation of plastics into the blubber of gray whale and seal. On effects on Arctic mammals, well, only the rat and mice data. And you can have a look at the word clouds on the type of effects that were shown. But even these studies are so far insufficient to rank endpoint sensitivities or predict no effect concentrations. On the occurrence in the Arctic birds, most studies were on northern fulmar, naturally, because that is also an OSPAR indicator. These indicator studies have been harmonized to plastic particles which are larger than 1 millimeter. And looking at the data sets available, we have the suspicion that that may be led to fewer studies on smaller microplastics, something to keep in mind when making guidelines. What we also see that the uptake of plastic seems to depend on the feeding strategy and the digestive tract, because some birds have extra stomachs and pouches where plastic can get stuck. For example, the fulmar has a gizzard designed to digest squid beads. Doesn't work as well with plastic, and the plastic has a hard time getting out of there. But also other birds have high frequencies of occurrence, and this is an expression often used to indicate the amount of individuals in a population who have plastic in them. And a new study on little auk showed that there was 100% of the individuals had plastic in their gula pouches. Pursuit diver birds seem to ingest less plastic. There are many more bird species that have frequencies of occurrence above 25%. Some of them are also used as food. On the effects on Arctic birds, we can only assume that large amounts of plastic in the stomach slow down food digestion. But there's some evidence from Australia on flesh-footed shearwater that correlated plastic and fibrosis in stomach lining. And plastics was found in spleen, kidney, and another digestive part, the proventriculus. And that was associated with tissue damage and inflammation. And in Japanese quail, even effects on body mass, growth rates, oxidative stress, enzyme activity changes, and endocrine effects, that means effects on the hormone system, were shown. On the occurrence in Arctic fish, 12 marine and 4 freshwater species have shown to have plastic in them. 1,627 Atlantic cod have been investigated, all of them mostly in the gastrointestinal tract, which for fishes only gives a snapshot in time. We would be interested in studies showing plastic occurrence in tissues. There's one study that shows plastic occurring in muscle tissue of Arctic shark and lake trout. The plastic was present but did not bioaccumulate in this study. Overall, we have to say the studies have low comparability because studies with investigating larger microplastics, like above 0.5 millimeter, had low frequencies of occurrence of 0 to 18%, while studies including small microplastics had higher frequencies of occurrence, up to 100%, pointing towards that maybe the results of the studies reflect more the methods than the actual conditions in the nature. Recently, there are more recovery tests that is methodologically very positive. Regarding the effects, take-home message, short exposure times. With microplastics, there were no effects or limited effects found. Short exposure times with nanoplastics found a stressed immune system, but the studies with a long exposure time, and here we are talking several months, which is expensive in a lab, and microplastics found a whole array of effects. Occurrence of plastic in Arctic invertebrates have been shown in bivalves and whelks, which is a kind of snail, amphipods, crabs, and starfish. And in Arctic blue mussels, there was an attempt in a nice review paper to sort them according to lowest observed effect concentrations. And the two first categories might be environmentally relevant, found some effects. And the middle category with many effects is maybe a bit higher than normally in nature, but still could occur locally. And effects on other Arctic invertebrates were shown in copepods, isopods, amphipods, and daphnia. With varying environmental relevance of the concentrations. But this Norway lobster study exposed for over 8 months with 5 fibers per feeding, which is probably not exaggerated, and resulted in a reduced feeding rate and lipid storage. So we came up with the knowledge gaps and recommendations for the future, and we really think that the effects of microplastics should be investigated in concentrations and combinations that are actually found in the environment, and both in individual animals, but also in the whole ecosystem. And effects of multiple stressors need to be investigated because there are results that show that plastics combined with other contaminants can have other effects than each contaminant by themselves. And there are other multiple stressors such as temperature, pH change, and so on that could play a role. So also we would like to have definition of biomarkers that can be used in a harmonized way. And I think I'll stop here. Thank you.
Thank you very much, Tanja. We'll go to the next speaker, which is Bonnie Hamilton, and she'll talk to us about occurrence and effects of plastic additive chemicals on Arctic biota. And Bonnie is an ecotoxicologist and postdoctoral fellow at the University of Ottawa. Her research integrates principles from aquatic ecology and toxicology, environmental risk, and conservation biology to investigate questions about the environmental transport, ecological fate, and effects of emerging contaminants under a changing climate. And she served in the AMAP expert group for the last 5 years, and you've recently been appointed to the Canadian Committee on Antarctic Research, and that's led by Polar Knowledge Canada. And please, Bonnie, it's up to you now.
Thanks, John, Rene. Hello, everyone. Hopefully you can see my screen.
Yes, we can.
So we're gonna switch gears and talk about the occurrence and effects of plastic additive chemicals in Arctic biota. And this is done with a variety of people from across the Pan-Arctic, and it's been a joy to lead this chapter. So we've heard a lot about the physical effects of plastic pollution, and from a range of sizes, all the way from micro, and most recently with micro and nano from Tanja. And now we're going to talk about another aspect of plastic pollution, which are those plastic additive chemicals. And plastic additives are added to polymers during the manufacturing process, and they're not often chemically or covalently bound to the polymer backbone, meaning that they can easily leach out and move into the environment, and so move from those plastic products into their surrounding environments. And so recently, a group led by Martin Wagner published the Plastchem report, which if you haven't had a chance to look at that yet, I highly suggest you do if you're interested in this topic. It's an extremely in-depth sort of state of the science of what we know about additives very broadly, with some really nice infographics, which I'm going to share with you now. And so when we think about the plastic cycle, we have feedstocks, raw materials, bulk materials, products, and waste. And so just to orient us, these black arrows are the flows of chemicals. These blue arrows are the flow of material. And so when we look at these plastic chemicals, we have our non-intentional added substances, which are added throughout unintentionally added throughout the process of producing and using and recycling plastic products. And then we have a variety of other substances that are added at different stages of the life cycle. And when we think about what this means in terms of what are plastic additive chemicals, it's extremely complex, let's say. There are lots of different chemical types that make up plastic chemical additives, like we see here with those different uses, which we're just going to dive in here. And so this wide range of chemical types, including organic chemicals like our organophosphate esters and halogenated compounds that fall within our organic chemicals, as well as inorganic chemicals. These chemicals make up a broad family of compounds with many different uses, including colorants and processing aids and plasticizers and stabilizers and flame retardants. And so when we think about this and zoom out to the 10,000-foot view, with over 16,000 chemical additives on the market to date, it serves as a very large, complex category of chemicals with many foundational knowledge gaps. And so when we shift to looking at the Arctic, and when we look at environmental compartments that have been assessed for plastic additives across the north, we can see here, based on the squares, the compartments identified with these squares here, there are really only a few environmental compartments, both abiotic and biotic, that have assessed plastic additives, a very small number of plastic additives, in these compartments, especially when we compare it to what we know about microplastics and those physical particles which we've assessed across the north in nearly every environmental compartment which we have found them in. And so this begs the question of plastic additives, again, are a little bit complex, just like microplastics are complex. And so when we look at an example from Nunavut and looking at seals in Nunavut, we've found that while seals have negligible amounts of accumulated ingested plastics, we have found a high abundance and occurrence of plastic additives in their tissues, which leads us to a few hypotheses. So I know this is a little bit messy, but we're just going to walk through this quickly just to kind of orient ourselves. So these gray arrows are plastic additives. These black arrows are physical plastics. And so when we think about how seals could be exposed to plastic additives, we have small zooplankton ingesting microplastics, which are carrying additives, right? Because those additives are part of those plastic particles, which can then leach out into the environment. And those microplastics and additives can be passed to the predator. Fish may directly ingest microplastics. They may also be directly exposed to plastic additives, those chemicals that are a little bit more water soluble, for example. And then those plastic additives and microplastics can be retained in fish. Some are certainly excreted. And then when it comes to the seals, the first hypothesis is, well, microplastics and additives are passed via prey. A second hypothesis is maybe those seals are directly ingesting microplastics and retaining those additives and excreting the physical particles, and that potentially nanoplastics are being translocated into the tissues from the gut of seals and carrying those additives. And so this is really just to show the point, and the fourth hypothesis is it could be a mixture of the above. And really what we're trying to communicate here is that it's actually quite complex. We've got those physical particles and those chemical particles. And so when we look at this as, well, what does this actually mean in practice and in the environment? We know that particles and microplastics can undergo long-range environmental transport as well as regional environmental transport from local sources. And those particles and additives are then leaching into the environment, which then leads to ecological exposure and effects. But then when we really start to think about what does that look like and what does that mean, we have a lot of questions, right? Are those additives hitching a ride and being transported from microplastics into the Arctic? What types of additives are undergoing that types of transport? What other additives are maybe being transported through physical pathways like the atmosphere and like ocean currents for those that are more water-soluble compounds? And so again, we have a lot of questions, which is great for us because then it leads us to asking and positing that question of what do we know about the occurrence and effects of plastic additive chemicals in Arctic biota, and what can we learn from the information that we can gain from the literature in order to recommend and push future directions in a coordinated way to assess chemical additives in the North. So very few studies exist. I'm actually just going to go back a stage here just to keep this on the slide for a second. So ultimately, we conducted a systematic review focused on a selected group of intentionally added chemical additives. And I just want to make a point here and emphasize this, that we did not prioritize regulated chemicals like PBDEs or dechlaranes, short-chain chlorinated paraffins, those chemicals that are under the mandate of the POPs expert group for AMAP. That group does a lot of excellent work. It has a whole body of literature focused on POPs. And so we focused not on those chemicals and just on the chemicals that don't fall within the POPs group, but also are related to plastics. So there are a lot of gaps. Very few studies exist, relatively speaking, on the effects of plastic additives in Arctic biota. And so of the studies that met our inclusion criteria, only 10% identified studies focused on the effects of plastic additives in Arctic relevant biota. The vast majority of them were focused on occurrence. And there were even fewer studies that looked at experimental exposure. So when we talk about what plastic additives mean to northern wildlife, there's a big knowledge gap. And so of those observed effects within those few studies, we see impacts on growth, oxidative stress, community structure, and transcriptomic responses. And so ultimately, we've identified several research gaps. And what you see on the screen here is a broad brush stroke of those gaps. There are several more, and they're quite specific in the report. But very briefly, we found that there is uncertain sources and transport pathways. So we know they're there, but we also have a lot of questions about how they get to the north and what the role those plastic particles have in transporting those additive chemicals. We also have these poorly characterized chemical profiles in terms of, quantities and polymers of how they leach under Arctic conditions. And there's quite limited data on exposure, bioaccumulation effects on wildlife and ecosystems. And so this led us to ask some questions about future directions and recommendations. And so ultimately, one of the big things that we discussed as a group is that we really need a clear definition of what plastic additives are to help guide research priorities and management decisions. We need more investigations on transport pathways to address data gaps and monitoring needs, but then also to really use screening tools to understand what is present in the Arctic to help inform more coordinated monitoring efforts. Because again, there are a lot of plastic additives and we certainly can't monitor them all. So we need to understand what's present in the environment in order to move forward in a coordinated way for environmental assessments and biological impacts. So I think I'm just going to stop there and then we'll move on to questions. Thanks.
Thanks a lot, Bonnie. And the structure here is that we'll just wait a bit with questions and answers, because as the first thing here, we'll be asking Eivind Farmen. He's the senior advisor from the Norwegian Environment Agency, and he's also one of the co-leads of the Litter and Microplastics Expert Group. And as I explained in the beginning, this is work that is reported through AMAP in a summary for Policymakers and a technical report to be published soon. And Eivind will take us through the summary and outlook recommendations that have been developed through this work. So please, Eivind.
Thank you very much, Jan. So first of all, I'd like to a big shout out from me to the experts in our expert group for making this systematic literature review. I don't think actually they mentioned that very clearly, but that's what they have done. Systematic literature review, which is very timely and well needed. So also thanks a lot for your presentations earlier today. And I would like to start my little summary by saying that even though I've been a part of this group now for some years, actually some of the findings did actually surprise me. I think the reason why I was surprised was because my impression from all the photographs you see of seals or birds entangled in fishing net, and all the popular science articles in daily media about negative impacts from microplastics in humans, and also knowing that there's an exponential increase in plastic research papers out there. All of that made me believe we knew a lot about negative effects. But then our report mainly shows that when it comes to Arctic animals, we don't know nearly as much as you think. I mean, for entanglement, as Ingrid showed us, there are some obvious negative effects, but the problem is that these effects have not been systematically investigated. So we don't really know which species is most frequently entangled or which species is most impacted by it. And then for ingested plastics, as Amy told us, although plastic particles have been found in many of the animals that have been studied, the majority of Arctic species have not yet been examined for plastic ingestion. And then, as Bonnie said, in some of the species, we do know a lot when it comes to the ingested plastic burden. For instance, the Nordic fulmar. But we don't know so much about the other side of the equation. That is the effects. That was Tanja's presentation. And then also we do have some lab experiments shown by Tanja. which with species that are relevant to the Arctic, but most of them have brief exposure durations and conditions that are not always environmentally relevant. So we don't have sufficient data to rank species sensitivities or endpoint sensitivities, and also too little data to derive predicted no effect concentrations. So my take home of the key conclusions is that we have a bunch of researchers that conclude that we don't have enough data. I guess you heard that before. But actually, I think this message is very important, because we do know that many Arctic animals are sensitive, as well as already under pressure from other stressors, such as climate change. So more knowledge about how and when plastic pollution affects Arctic animals should actually be a priority for Arctic nations. So I want you to use our coming report to go home and tell your research funders, because it does show or direct for further studies that is needed in order to get closer to really understanding the negative effects. Okay, so and what's our outlook? What do we need in order to get closer to this assessment of negative effects? As I read the report, the answer from our authors is that we should build on what we have. We already have some research and monitoring in the Arctic, and our experts have recommended that sampling and data collection need to be strengthened and better aligned. And they also highlight opportunities found within indigenous-led community-based monitoring, simply because we rely on local settlements in order to observe these things, because the Arctic is very sparsely populated and with large distances to cover for research and monitoring. And then more specifically, we need harmonized and interoperable databases and protocols for entanglement observations. And we need controlled long-term effect studies with environmentally relevant concentrations and types of microplastics. And also we need improved understanding of the process for leaching and or chemical additives and their environmental presence. So with this, I would like to thank all the contributing experts again. Our expert group will now be starting to make preparations for a future assessment of litter and microplastics in the years to come. And we will do that by starting to look at where and how to harvest data, what kind of data we do have that are comparable, what kind of data laundry that we need in order to make data more comparable, etc., etc. So stay tuned, and I'll hand over to young Rene for questions. Thank you very much.
Thanks a lot, Eivind, and I would like to follow you in thanking the presenters today and thanking the expert group as such. It's been a long travel to this point in time where we are now, but the report will soon be ready to be published. And as Eivind said, we're looking forward to preparing for assessment work in the coming years. And should there be someone in the audience who would like to contribute, there is also a process to nominate also from non-Arctic states to the expert group. We have time for some questions, and the way this should work is that you as webinar participants should be able to raise your hand, and when you do that, I should be able to see that here on my screen and can give you the floor, and you can raise questions to the experts here today. So for the participants, any questions or comments that you may have to the work, please go ahead and raise your hand. Yeah, Marte Have, and I'm now pushing a button here which should allow you to talk. So please, Marte Have.
Hi, and thank you for a great set of presentations. It was very interesting. One question, the most pressing for me is, do you have any recommendations for us to what chemicals that you would like to start monitoring? You have a list of priorities that will be given as well. Thank you.
Bonnie, would that be you?
Sure. Yeah, that's a great question, and it's something that we talked quite a bit about as a group, and I'm not quite sure we're there yet. Kind of what I was trying to get at toward the end of the presentation is that we know, like UV328, for example, would be an easy place to start. And some of those UV filters and stabilizers would be a good place to start. But again, I think we need to do a little bit of screening of what is in the environment to help guide us in putting forward those recommendations. And there's been some discussion as well of LMAG, this plastic litter and plastics expert group, doing some cross-pollination, as you will, or coordination with the POX group as well to understand sort of what we know about some of those chemicals that kind of straddle the line between solely plastic additives and persistent organics, because plastic additives kind of span that entire spectrum. So ideally, it'd be really great if we could provide, a full list of chemicals that we should be monitoring, but I'm not quite sure we're there yet.
Thank you.
Mm-hmm.
Other questions, comments to the presenters? We can close the webinar for today. Thank you to the participants for your interest in AMAP's work. We'll be preparing other webinars like this on other matters related to the AMAP work program. Please keep an eye on the website, but also we are on social media where we announce these webinars. And again, should you be able to or interested in contributing to AMAP work, also please contact us. We have a process for inclusion also of non-Arctic state experts in our work. But again, thanks a lot to the speakers today. Thanks to the webinar, and I'll close the webinar now. Thanks a lot, and bye-bye.